So in the last video, we talked about how we had redundancy, which served as a way to protect ourselves against changes that could cause problems for us. However, it's important to note that sometimes changes will occur no matter what. We call the change in that base pair sequence of DNA a mutation. And while many of these mutations are totally harmless and don't have any effect, some of them can be helpful and lead to new, exciting traits, but many of them can be harmful and even fatal. However, even though there is some harm or fatality involved with mutations, mutation is the mother of all change and is absolutely essential for evolution to occur. If there were no mutations, then we would not see the different variety of people or even organisms in general that we see today. Now, it does matter which cell type you are when looking at mutations. If we look at somatic cells, which are body cells, the cells that make up your skin and your liver and your lungs, these cells cannot be passed on to your offspring. That doesn't mean that this is harmless, because many of the mutations that happen in somatic cells can cause things like cancer. Cancer is a collective term for cells that have just kind of gone haywire and are replicating way over the top. But our immune system do have a way to check them, and cancer as a diagnosis only happens when your immune system failed to stop it. The other cell type are gametes. Gametes are going to be the sex cells, sperm and eggs, that do get passed on to offspring. Now, many mutations cause no damage, but others can cause birth defects, and even worse, others can cause miscarriage or loss of the pregnancy. The most damaging types of mutations are insertions or deletions that occur near the beginning of a gene, because then everything downstream will be shifted. So if you add in a base or you take out a base, you've completely shifted those codons, since we're working in groups of 3. We call this a frame shift mutation, since every single codon down the line will probably be affected. Since ribosomes can't actually read the mRNA strand, they understand the nucleotide alphabet instead, they can't actually account for this frame shift. They can't fix that. So instead, they read every 3 codons, regardless of whether or not those were the original codons or not. So let's look at this. Let's look at insertions and deletions versus a substitution and see how it changes our DNA. So we have our normal DNA strand, we see our mRNA molecule we get out of it, and we see our normal protein. If we just substitute, meaning we swap one base for another, in this case we've swapped out an A and added in a T, we see that we've only changed one amino acid from the original, right? Met, cis, ser, gly, arg. And down here we've swapped out cysteine for a serine. So one single amino acid changes if we substitute one base for another. But if we look at an insertion or a deletion, we see that everything from that point down has changed. Instead of serine, glycine, arginine, we see glu-arginine lysine. Or we see alanine, glutamic acid. So the entire protein downstream is completely different. And this can be much more harmful, since the actual entire protein gets changed, not just a small little piece. Mutations like that can lead to illness, because the protein is very reliant on the shape in order to do its job, and the shape is reliant on the amino acids present. So if you change the amino acids present, you change the shape. And if you change the shape, you may end up with a protein that does absolutely nothing. So if the protein or enzyme is supposed to look like this, but the change in DNA and RNA leads to a protein that looks like this, we can no longer do the job. In this case, it's an enzyme, and since we've changed the shape of the enzyme, we can no longer actually speed up that chemical reaction. In this case, the reaction is taking down and breaking down something toxic. And if we're not able to break down that thing that's toxic, that toxic waste builds up. So we end up with this high level of some toxic chemical in the body that can cause sickness or even death. So, for example, Tay-Sachs disease. Tay-Sachs disease leads to a buildup in the brain, which then it does lead to fatality within 3 to 5 years. So most people who are born with Tay-Sachs disease will not make it to kindergarten. So sometimes the protein is misspelled, so to speak, meaning the sequence of the amino acids is incorrect. And if we totally change, if we change that enzyme even in a small way, that can cause the enzyme to no longer work. So we see that here. We see that we have this normal protein functioning. It's forming this enzyme called phenylalanine hydroxylase, which takes phenylalanine and converts it to tyrosine. And so we see that we have phenylalanine and we push it through our enzyme, we get tyrosine at the end. But in the non-functioning side, our phenylalanine cannot actually fit. And since it cannot do that, phenylalanine never becomes the tyrosine. A lot of different diseases and physiological problems are because of this, because some sort of enzyme has stopped working or never worked in the first place. So overall, mutations can be very dangerous, even deadly, but at the same time can lead to amazing change. DNA is really our basis for all the things that make us us and make us different from each other. It's something that both unifies and defines us as individuals.